So in this presentation, we'll be going to talk about the aspects of the genomics, and mainly we'll be focusing on the gene finding and the genome annotation approach. So first of all, suppose we are going in the detail of the genomics, then we have to understand that how this genome word has came into existence and how this genomics has came into existence. So all change utterly, a terrible beauty is born. A world of bacterial genome sequencing changed utterly when the first two complete bacterial genome sequences were published by the scientists at the Institute of Genomic Research, Tiger in Maryland, 1995. So in 1995, first time the whole microbial genome sequence has came into picture, and due to that only, a new science has born, and that is known as the genomics. And from that only, we also had the development of lot of other omics techniques. So we'll be just observing that how this science of the omics is working. So first of all, suppose you want to know the meaning of the word omics, then what omics is. So this omics term, it symbolizes a redefinition of how we think about biology and working of the living system. Suppose you compare this initial upper line, then you can show that when you're having the limited number of the tools, we'll be able to create a small houses or a limited thing. Same is true with the biology. When we are having a very limited tool, we were not able to interpret the entire details of the biological science, and very few hypotheses were into picture. But after getting the new science of the biology and after getting the new techniques like the sequencing, microarray analysis, and lot of other, we were able to build a big house. And similarly, we are able to also find out the new hypothesis and new concept of the biology. So that is the basic meaning of the omics. Initially, the word is popular known for the genomics only, but after that only this omics word has become a suffix for so many different kind of the science. And right now, whatever you are observing, that is only the tip of iceberg. In the coming days, you will see such kind of the number of the different genomics, proteomics, transcriptomics, and lot of other omics science in the biology. So that way, science is now smartly moving towards the science of the biomics. Why so many omics are there? That if you're having only one tool hammer, then you will just have the one thing that you can just tell the need. But when we're having the advanced tool, you'll be able to do so many things. So the same thing is happened with the biology. Initially, we had a very limited knowledge of the biology, and we had the only the fundamental principle like the Darwin evolution principle, Mendelian genetic principle. After that, only in the 1953, slowly the DNA modeling is came. After that, only we are able to find out the structure of the protein, and from that only, slowly we had that concept that all the biological molecules they can have a specific structure and they can also have a specific sequence. And from that only, with the help of the different computational tool, we have developed the different databases where you can store the information about the structure and information about the sequencing. Similar to that only, whatever the computational method has developed, that computational method is also developed the different kind of the algorithms and different kind of the techniques. And due to that, now it is possible that instead of going for all the wet lab exercise, from the data which is generated from the next generation sequencing or any other kind of the advanced technique, it is possible to use the different biological tool mainly in the computer, and we can find out so many new things out of that. And that includes the structural genomics, computational genomics, comparative genomics, functional genomics, and so many other. So that is the entire world of the omics. So we had the development of the computer and internet also along with the development of the biological science. Precisely, suppose you want to see that how this omics is working, then you can just have this comparison that for example on this line you can just find all the om. Suppose you want to define the genome, then genome is a complete set of genomic information of any cell that is normally known as the genome. Same way, transcriptome means it will cover all the different kind of the RNA. Proteome means it will cover all the different kind of the protein. Same is true with the interactome, functome, and textome. When the simple holistic information, it is joining the sequence, structural, expression, pathway, regulation, and network with the help of the different bioinformatics tool. This will be creating the one matrix that suppose I'm just merging all this information into the genome, and that will create a science of the genomics. Suppose I'm merging it with the transcriptome, then that will create the transcriptomics. Same way, it will also create the proteomics, integromics, functomics, and taxonomics. So that is the second kind of the development of the science, where you are having the two matrices. One is the biological science, and second one is the information science. So that is a way by which normally this genomics and other part is working. Suppose technically we want to give the definition of the genomics, then genomics is the term which is coined by the Thomas Roderick in 1986. It refers to the mapping, sequencing, and analysis of the genome, and this definition have enlarged to include the genome function also. So that way, in broad way speaking, genomics is the science which is dealing with the entire details at the level of the genome. So that would be the fundamental part for the genomics. Suppose we are taking the genomics exploration, then basically what will happen in this? And suppose we have learned the different kind of the NGS sequencing technology. By any kind of the sequencing technology, whatever the genomic information after the sequence of the genome you'll be getting, that will be simply like the sequence of the ATGC. For example, this is the physical map of the HIV virus. You will see that this will be only the ATGC. But this text is not giving any meaning to us. We are not able to find out that how many genes are there, how many genes and what are the function of that gene, from where it is gene is getting started, where it is getting ended. So we are not having any biological information to that. So this is known as the physical map of the any kind of the genome. For example, we have taken the physical map of the HIV virus in this slide. Now suppose you observe this second figure, then you will just find out that as a biologist, we are able to understand this figure. We can just probably find out that this is the entire genome of the HIV that will have these three gene, gate, pole, and envelope. This is the starting position, ending position. This is the RU5, this is the U3 region, and this is the length of the gene. So such kind of information, when we are adding to the basic information, then that is known as the genetic map. And as a biological student or as a biological scientist, we need to convert the information of the physical map into the some human readable kind of the genetic map. So that is the biggest task for the biological scientist, and even now that is done with the bioinformatics. Now suppose you are taking the different concept of the genomics, then you can also have the three types of the genomics. The first type of the genomics, that is known as the structural genomics. So that includes the DNA sequencing, that will include the mapping of the gene, and that will include the annotation process. So that is the first kind of the genomics, which is known as the structural genomics. Now suppose you are taking the second part, that we want to find out that what is the function and what is the expression of the gene under different conditions. So that will come as the functional genomics. So that will include the expression profiling, that will also include the transcriptomics and proteomics, and that will also include the analysis of the mutants. So that will tell about the function of the genes in a different condition. And thirdly, suppose you're having more than one genome sequence with the related species or with the different kind of the strains, then you can also go for comparing the entire genomic study of that. So that will come as the comparative genomics. In today's lecture, we'll be covering about this structural genomic aspect. We'll be talking about the mapping of the genes or gene identification and some kind of the annotation process, which is normally utilized in the practice. Now, suppose you are going for the simple steps of the genome analysis, then genome analysis steps are going in this way. The first step always is to go for the data quality and checking and pre-processing. So whatever the data is coming out from the NGS platform, that will have the different level of the quality. So in the first step, that would be checked, and after that, initial pre-processing will be done depending upon the kind of the NGS data you had. The second part is that normally in all the NGS sequencing data, we normally going for the fragmenting the DNA. So whatever the data we are getting, that would be the sequence of the fragment. So from that only we have to go for the assembly of the sequences, and we have to find out what are the markers for that. Then the next step is coming in the case of the eukaryotic, lot of repetitive sequence would be there. So you have to mask out and filter this repetitive sequence. So this repetitive sequence is like that. That for example, when I'm taking the lecture, I'll be using some of the word repeatedly, even though that words are not having any kind of the meaning, but due to my habit, you can see that that words are getting repeated. And from that only, you can just make out that this is my text or this is the lecture which is taken by me. And suppose you want to reduce the information in my lecture, then what you can do is you can remove this kind of the words which I'm using repeatedly without any kind of the function. Same way in the case of the genomes of the eukaryotes, there would be a lot of junk DNA. So that is the non-coding region and that is getting always repeated. So this third step is to identify such kind of the repeated sequence and to mask out it or to filter it. Then the next part is that we have to find out that what is the location of the gene, and depending upon only we have to find out the genome mapping. So we can just find out that in given genome I'm having the presence of the 5 gene and the one number gene that is starting from example one nucleotide and it is going up to the 200 nucleotides. So like that only physically you can mark out that what are the positions where your gene is getting started and where your gene is getting ended. The next step is that is the gene prediction, and you have to train the model for each one. So whatever this gene prediction is there for that, the different kind of the models is available, and depending upon that only computationally it is possible to go for the prediction of the gene. Then you can also go for the genome analysis, specifically when you're having the data of the EST, express sequence tag, and the cDNA sequences. You can also go for the transcriptomics like study, means the expression under the different conditions. So that will come under the functional proteomics, functional genomics only. The next part is that genome annotation, that whatever the gene you have predicted, we have to find out that what is the function of that gene. So that is known as the genome annotation. And functional genomics under the different condition, how it is acting, that would be studied. And in the comparative genomics, you can just compare your genome with the other available genome or with some other genome of your interest. So that would be the different steps of the genome analysis. Now we are starting with the very first part, that as I told earlier, that whatever the genome sequence we are getting, that genome sequence would be also having the data quality. So whatever the data are coming, that is of different quality. So you have to select the quality of the data which is good. So you can just have the condition like this, and this is typically known as the Fred. So you can see that you can have the three level of the data. One data which is known as the high quality data, in which there is no ambiguity. The data is quite precise and it can be readily utilized. The second kind of the data is that is the medium quality, and that is having the somewhat lesser kind of the quality, and that is somewhat ambiguous also. And the last one is that is the poor or the low confidence data. So you can just assume the condition that, for example, I'm taking the lecture in class. Depending upon my voice, whatever I'm speaking in the class, you can see that the persons who are sitting on the first row, they are able to understand and they are able to write my lecture precisely, as quality of my voice was reaching to them, that is quite good. So suppose you are taking the notes from the person who is sitting on the first bench, the quality of that would be quite good. Suppose you are going in the middle of the class, then what will happen? In the middle of class, from the entire lecture, suppose I'm speaking some sentences in a low noise, they are not able to understand, or they'll be missing some kind of this word or some sentences. So the quality of the data which you are taking from the middle level student or the middle bench student, that would be comparatively low. Suppose you are taking the last bench student, then what will happen? Suppose my voice is not big enough, then what will happen? The person who are sitting at the last, they are not able to understand my lecture completely. And when they are writing the notes would be having a very lesser information. So that is known as the low confidence or the poor data. So same way, whatever the sequencing data would be there, that would be also having these three kind of the quality. There are computer softwares which are available that will just filter out the quality of the low confidence, and that will just keep the data of the good quality and medium quality, and even you will have the control of that. So when we'll have the practicals for that, we'll have the detailed explanation about that how it is working. But this would be always a first step, that out of the total data you obtain, we are removing the low quality data and we are selecting the data which is of good quality. The next step after that is coming, which is known as the genome assembly. So genome assembly is a complex process. In bioinformatics, sequence assembly refers to the alignment and merging of the fragments of a much larger DNA sequence in order to reconstruct the original sequences. This is needed as DNA sequencing technology cannot read the whole genome in one go, but rather small pieces between 20 to 1000 bases depending on the technology you use. Normally, suppose you are taking the childrens who are just learning that how to speak, and you will see that when the teachers or when at home they are just trying to speak, what they will do, they will not speak the entire sentence. They will initially speak only few words, right? They will just learning that how to speak mom, dad, I want to, I don't want, such kind of these initial small segment they will learn. After that, they will just join all these sentences together, and then they are able to make out the full sentence. Same way, whatever the genome sequencing we are getting, that would be having the smaller words like this of 20 or 1000 bases only. So the first step for us is we have to stitch all these things together and we have to create the entire part. So typically a short fragment are called reads, and from that results from the shotgun genomics and DNA transcript EST. So this is normally known as the reads or the shorter length of the sequences. You can have the two approaches. Initially, when we had the Sanger sequencing and other, then we used to go for the cloning approach, and we are just taking the one plasmid-like thing. In that only, we are cloning the entire fragment of the DNA, and that is going for the sequence, right? Suppose you are comparing that I'm having the book. In book, I'm having the sum of the pages, and I'm just reading these pages. So that would be similar to that, reading the clone fragment of the DNA. Now, whatever the techniques are using in the NGS, what they're trying to do is they're removing the cover of all books, they're separating the each pages of this, and after that only they're also tearing off all the pages into the very small fragment of the, for example, we are taking 400 or 500 base pair only, and all this would be merged together, and after that we try to read it out. So that is basically known as the whole genome shotgun assembly. So whole genome shotgun means whatever the sequence is there, that is randomly break from the different part. And now the task is becoming somewhat complex for it. That whatever the tier of paper is there, from that only you have to recover the entire story. So it is condition is like this. That for example, this is known as the thresher. So I've written something in this paper, and after that I'm just going for tearing it off. So you can see that over here something is written, but that is getting teared off. Now suppose from this I want to recall that what is written, then what I have to do is like the normal puzzle game, we are just solving the puzzle. We have to arrange all the fragments of the paper, and depending upon the joining of this paper only, you will be able to find out that what was written on this paper actually. So the same kind of the thing is done in the case of the genome assembly, right? So you can have the another explanation for that. That for example, I'm having the sequence fragment DNA. I'm having the one fragment which is shown black in color. I'm having the one which is shown red in color. Again, I'm having the fragments which are black, yellow, like this only. Now these are the short fragments. You can see that that is getting only the piece of the information. Now suppose below to that you are getting the second kind of the fragments. It will be having the mixing of the two. That for example, this is having the half portion of the black and half portion of the red, right? Same way there are other sequences where the larger fragment of the red is there and smaller fragment of the black is there. Same way it is other. Suppose I'm just combining the information of all of this, then I can find out that when I'm just removing all the duplicates, I'll be able to generate the counting, and this contact would be having a larger length with comparison to the initial sequence. And I'll be also confident that whatever the information is coming in between, that is also marked at the 4 5 places. So the condition is like that, that suppose I'm writing my name Rajesh Patel, right? You are knowing that Rajesh is my name and Patel is my surname. So this is the entire thing which I've written in one paper. Now suppose I'm writing the Rajesh Patel for 5 6 times, and after that I'm cutting this from the different part. For example, I'm cutting it the from the Rajesh. After that I'm cutting with the Patel. Over here the second cut I'm creating that instead of getting the Raj I've cut the Raj, but after that whatever the ESH is there that I've kept, and after that PAT I've kept. Like that only you can just recall that all the different level of the fragments I've generated. So that may have the some words of the Rajesh and some words of the Patel. But when I'm merging all the information together, I'll be make out the entire name Rajesh Patel out of that. So that way this assembly is done. So you can see that this is the one kind of the config, and this is the config in which we are not getting any kind of the thing. So that is a way by which normally assembly is done. So the same kind of the example is there. You can see the entire thing. and whatever the masked DNA sequence you have seen earlier, so that process is given over here, that you can see that whatever the sequences are shown in the brown color, so these are the sequences which are getting mask sequences or the repeated sequence. So that also you can remove, and you can generate the config out of that. So that is a way by which the config or the assembly process is done. Suppose you are observing the real life example that how it is acting with the ATGC, then you can see that this is the fragment ACCGT, this is the CGTGC. So these all fragments are there. Now when I'm aligning all the sequences together, I'll be just getting the sequence which is a unique sequence to all of this, or the consistent sequence which is common in all of this. And you can see that this A is coming at the 1, 2 and 3 places. This C is coming also at the 3 places. This C is also coming at the 3 places. And we are just able to add the information on this, that is from the TT side and GC side. So like that only we'll be able to create the entire information into the one kind of the fragment. So this is a way by which the assembly is done. Same way you can see that how the assembly is created, that is shown in the another kind of the figure only. Now for assembling this, we are using the different kind of the algorithm. Whatever you have seen, that was the concept by which normally assembly is done. But here are the some of the softwares which are utilized. So the Tiger is the one software which is available. FREP is also there. Calera is there, then Cap3 is there, Reset, Fusion, lot of many softwares are there. Typically, we'll be taking the example of the Cap3. Similar to that, each software will have their own kind of the logic to compare the sequences, to assemble the sequences, right? In Cap3 assembler, what will be done? First of all, we'll be removing the poor and the regions of the read. So whatever the fragments are there, depending upon the quality of the data, the poor and the regions of the reads are removed. After that, computationally, we want to find out that what are the overlap between the reads. So whatever the fragments are there, these fragments are getting some kind of the repeated things or not. So that would be done. We are removing the false overlap, and after that only we'll be constructing the context, and we'll be just have the construction of the multiple sequence element and generation of the consistent sequence. So this is a way by which it is done. Now step 3, some of the other details, that for example we are observing that in this I'm having the one sequence over here, I'm having the second sequence, I'm having the third sequence. So for example I'm having the H, F and G. Now you can see that each sequence are having the some portion which is getting overlap, and this portion is basically known as the clipping position. So you can recall the panoramic mode in your camera. So in camera, we are having the one mode which is known as the panoramic mode. When we are operating the panoramic mode, what will happen? You will have the one picture. After that only we are taking the second picture. But when we are taking the first picture and second picture, you can observe that at least 10 to 20% of the frame that is getting again repeated. And this kind of the repeated will be occurring for the each snaps whatever you are taking. And after that only, when your entire 5 or 10 shots whatever your camera has set, that is getting over, the camera will take some time, and in that only it will computationally stitch all the images. So whatever the overlapping images are there, that would be merged with each other, and will be generating the one big panoramic view. And in the panoramic view you can see that a larger view with comparison to the normal camera photograph. Same way you can observe that this is the region which is getting overlapped. So that is known as the 5 days clipping position. This is known as the 3 days clipping position. And depending upon that only, the information will be repeatedly merged and you can see the comparison, right? So this is the sequence config. You will have the small sequence fragments. From that only the super config is generated. So this is the one sequence config which is generated due to the process of the sequencing. So this is the process which is known as the initial step, and that is coming under the heading of the genome pre-processing and genome assembly. And after that, the next step is there, which is known as the genome annotation. That first of all, what is the annotation? So annotation is a simple process in which we are just adding the information to the any object. That for example, if you are drawing some kind of the figure, then after drawing the figure only, you are putting some kind of the name to that. For example, you are using the Google map, and in the Google map also, when you are just zooming in, you can see that whatever the objects are there, the name of that object or some other details will be written. For example, you are observing the VNSU map in the Google map. Then initially you can see only the VNSU on the map. When you are enlarging further, you'll be also see the department. So the building name of the department would be given. So that is known as the annotation process. So genome annotation is the process of attaching the biological information to the sequences. So it will include the identify the elements of the genome. It will attach the biological information to the element, and it will store the information in the different kind of the biological database. So that would be including the process of the genome annotation. For the genome annotation also, we are having the different kind of the steps. The genome annotation in the case of the eukaryotic, always it will have the first processing as we have seen earlier, that is the repeat masker or the repeat identification. Second very popular thing is there, which is known as the homology search or BLAST or FASTA. That for example, whatever the fragment of the sequence of the DNA I'm getting, I can just run the BLAST or FAST for that. And if similar sequence is already available in the database, I will find out that this is the sequence which is having this kind of the function. The next step is that even when you are getting some kind of the new genome, and for that you are not getting the similarity search of the blast and fast, then you will be not getting any result in this. And what you can do is you can utilize the gene prediction tool. Some of the gene prediction tool again we'll see in the later in much detail, but you can just right now remember the name of such tools, so that are the gene scan, grail or the grimmer. Even the characterization of the gene GCG tool bench is available, so that is also available commercially, that is utilized to go for the genome identification and gene identification. Now suppose you are going for the computational approach for the gene identification. So there are the different kind of the algorithms and different kind of the softwares which are available for the gene finding. So first of all, what is the computational gene finding? So given an uncharacterized DNA sequences, find out that which region codes for which protein. So that is the fundamental question we try to find it out. Which DNA strand is used to encode the genes? We are having the two strand. So out of the two strand, which strand is utilized? Which reading frame is used in the use strand? So in the each reading frame, as we have seen earlier also, there would be six reading frame, plus one, plus two, plus three, and minus one, minus two, and minus three. Where does your gene starts and where does your gene ends? And what are the exon introns boundary in the case of the eukaryotes? And where are the regulatory sequences if it is present in the gene? So this all kind of the things are normally included in the computational gene finding studies. Now suppose you are observing the basic difference between the prokaryotic and eukaryotic gene, then you can see that prokaryotes, so that genes are comparatively small. The eukaryotes, the genes are comparatively large. As we are having the space limited, there would be high coding density. In the case of the prokaryotes, we are not having any introns, and we are also having the multiple protein which is created from the one gene also. So that is known as the polypeptide approach. So that kind of the things are not present in the case of the eukaryotes. In eukaryotes, you can have the low coding density, and there would be presence of a lot of introns. Introns means you will have the non-coding region. It is like that, that if I am having a house of only one room, then in one room only I have to arrange my kitchen, in one room only I have to arrange my living room, and in one room only I have to arrange my bedroom also. Right? So everything would be quite near to each other in the case of the prokaryote. When we are coming to eukaryote, it is like that, that I'm having the fleet of around 10 rooms. Then I can utilize each room for the different purpose. Even some of the room I can without use also I can keep. So that way you are using the places which are available on the genome. Gene identification, it is relatively easy, and the success rate is around 99% in the case of the prokaryotes, whereas the gene identification with the help of the computational method, that is quite difficult in the case of the eukaryotes, and only 50% probability of getting good results are there. The basic problems which are coming in the prokaryotes is overlapping ORF, short genes, and some kind of the TSS and promoters. In the case of eukaryotes, we will have a lot of problems. So that is the basic comparison of the prokaryotic and eukaryotic gene finding methods. Basically, prokaryotic gene finding methods are quite successful in the science. This is the greater details of the eukaryotic genes. You can see that this is the one fragment of the DNA. In that only you can find out there are some yellow color portions. So this is acting as an intron. Introns means that is the non-coding region. So when the primary mRNA is created from that, that will get transcribed. After that, you can have the process of the RNA splicing. this introns would be removed and you will have the mature RNA. You can see that in this only coding region is coming, and that is normally going in the cytoplasm, and from that only we are generating the protein. So that way this complex process is present in the case of the prokaryotic gene. When we are coming to the eukaryotic gene, and when we are coming to the prokaryotic gene, you can see that the DNA would be only having the coding region only. So whatever the mRNA is created, that is directly going for the translation into the protein. So that is a structure which is observed in the prokaryotic condition. So for the gene finding, we are having the different approach. So the first approach is there, which is known as the similarity based or the extrinsic, which is use the similarity to annotate the sequences of the protein, cDNA, EST. So this is the extrinsic approach. The second approach is that comparative genomics align the genome sequence from the different species. And third approach is there, which is known as the ab initio gene finding or the intrinsic information. When I'm not having any information available in the database, then depending upon the information what I'm getting from the sequencing, I have to find out something out of that. So that is known as the ab initio approach also. Then we can also have the integrated approach. In the case of the integrated approach, more than one method details are kept together. So that is normally known as the integrated approach. We'll see one by one that how it is working. Similarity based method, that is comparatively quite simple. You will just go for the simple search with the help of the blast and fast, and you can find out that whatever the genes and proteins which are already in the database, and if it is similar for more than 50% or 90%, whatever your criteria is there, then you can identify the function of them. Normally, more than 50% genes you can identify by this technique. Depending upon suppose some novel organisms are there, then that would be less. Limit of the regions or similarity is not well defined. So that is the basic limitation of this method also. Then you can also have the nucleic acid characterizing or the recognizing the coding sequencing. So three general solutions to finding the gene problems are there, that all genes have the certain regulatory signal positioning in about them. All genes by definition contains the specific codon pattern, and many genes have already been sequenced and recognized in the organism. So we can infer the function and location by homology if our new sequence is similar enough to an existing sequence. All these principles can be used to help and locate the position of DNA and are often known as the searching by signal or searching by content or searching by the homology interference. So this is the basic methodology. Depending upon that, the gene finding processes are normally utilized. So the first thing is that what kind of the signal sequences are present. So normally you can have the two kind of things. One is known as the URF, unidentified reading frame. Any potential string of amino acid encoded by a stretch of DNA, and any stretch of DNA has potential URF for any combination of six potential reading frame that will have the three forward and three backward. You can have the ORF. So open reading frame, by definition, any continuous reading frame that starts with the start codon and stops with the stop codon, not usually relevant to the discussing the genomics of the eukaryotic DNA, but very relevant when we are dealing with the mRNA, cDNA, or the prokaryotic DNA. So it is like that, that suppose you are going to the railway station, and if you are observing the train, then you can observe that there will be some coach or some bogies. Always the train which is going in some direction, you will find out that the direction in which train is going, there would be engine, and the last bogie or the last portion of the train, that would be a coach of the guard or the bogie of guard. So depending upon the location of these two, you can tell that this is the one full train and it is going in this direction. So similar to that, entire open reading frame would be identified, and depending upon that you can identify the genes. Now, how to identify the initiation strat? That, as we have seen that in the train, we can identify that from where the train is going. So that will identify depending upon the position of the train engine. Similar to that, only in the case of the entire signal searching, we are having the start sites. So in the prokaryotic, we are having the prokaryotic promoter or the peninsular sequences. So this kind of the sequences are present wherever the start initiation site is there. In the case of the eukaryotic, transcription factor binding site database is there, and depending upon that, we can have the TF binding sites. We can also have the SD sites, and we can also have the COSEC sequences, and we can also have the AUG or the start codon in 90% of the genome. Exception is some prokaryotic or the organelles. So depending upon this kind of the presence of the promoter, presence of the binding factor, presence of the SD sites, presence of the Kozak sequences, or depending upon the presence of the start codon, we can mark out that this is the portion from which your gene is getting started. Then, as we have seen that at the end in train, we are having the guard coach. Similar to that, when the sequences are getting ended, normally you can have the nonsense or the chain termination or the stop codons. So these are the three stop codons which are present usually. That is the UAA, UAG, and UGA. You can also have the eukaryotic terminator consists, and you can also have the eukaryotic poly A adenylation site, but some exceptions are there. So depending upon the condition, you can have the different kind of the sequences which will indicate that this is the place your gene is getting ended. Now, even in the case of the eukaryotes, we have seen that there would be the presence of the exons and the introns. So introns are the non-coding region and exons are the coding region. So as between the two bogies of the coach, we are having some kind of the junction by which only they are getting joined to each other. Similarly, in the case of the exon, you can observe that some portion is there which is known as the donor site and some portion is there which is known as the acceptor site. So between the two exons, there would be one intron, and this intron would be having some kind of the sequence at the end. Depending upon that, you can identify that this is the starting point of the intron and this is the starting point of the exon. So this is the way by which even you can find out that the exons and introns in the genome. The next one is there, which is known as the content approach. So strategy for finding the codon region is based upon the content of the genome itself. So searching of by content utilizing the fact that gene necessarily will have the many implicit biological constraints imposed on their genetic code. This includes the certain periodicals and patterns to produce the distinctly unique coding sequence, and non-coding states do not exhibit this type of the periodic compositional bias, and this principle can be helped to discriminate the structural genes in two ways. So this is telling like that suppose you are having some kind of the text which is written, you can see that whichever the text is having meaning, that will have some kind of the pattern, but some of the text which will not have any meaning, that would be normally like jargon. The same way is there in the exam. Suppose you are very confident in answering the question, then you are writing precisely. Each of your sentence and each of your word will have the true meaning. But when you are not knowing the correct answer and when you are bluffing, what you will do, you will repeat the questions or you will repeat the some kind of the word without any sense constantly. So that way you can make out that the this kind of the things are written properly and this is the thing which is not written properly. So that is a way by which even we are analyzing your exam paper. When we are analyzing your exam paper, within 5 or 10 minutes, we are able to assess your entire answer sheet. So during this time, we have not read your entire question paper completely, but whatever your answers are there, depending upon the writing pattern, due to our experience only, we are able to find out that whatever you have written, it is logical. Truly, you are knowing the topic and you have written according to that, or you have just bluff only. So same way, depending upon the content, you can just find out the things. So this is based on the local non-randomness of the stretch or based upon the non-codon usage of the particular life form. So one part is there, that is the non-randomness, and second one is the non-codon usage. Non-codon usage means, as you have seen earlier also, that each person would have the specific kind of the bias towards the some words. For example, some words would be always repeatedly utilized in my lecture. Similar to that, even in each organism, out of the three triplet codon for the each amino acid, each one is not utilized in a uniform pattern. Some codons are utilized at a higher rate with comparison to the other. So same way you can just make out that whatever the fragments of the DNA you are analyzing, in that suppose you are able to see such kind of the codon usage pattern, you will be identifying that this portion is coding or this portion is not coding. So this is the content-based approach. So you can see that non-randomness technique. So you can see that this kind of the plots are generated and you will have some threshold value. So this will tell that this is the above threshold value and this is the below threshold value. So whatever the things are coming above to that, that is the portion that is coding for something. And whatever the portion is coming below to that, that is the non-coding portion, or that might be a introns in the case of the eukaryote. So like that only you will find out that non-randomness, and this is normally done in the GCG test code, right? So that is a commercial software in which you will get the analysis like this. The same thing is that it is done with the codon usage technique. So depending upon the codon usage also, you'll be able to find out that what are the fragment of the DNA which is coding for some meaningful thing out of that. Then you can also have the internet, World Wide Web resources. So a lot of softwares are available. Again, we'll have the practical for that. So at that time, we'll discuss in much detail. So these all are the softwares which are available freely. Some of them are also available freely on the net. So practically, we'll be using that also in our practical sessions, right? Homology interference, how it is done. In the homology search, as you've seen earlier, normal it is going for the blast and fast, and depending upon the comparison it will tell that this is the fragment of your DNA which is matching with the another fragment, and this is the function of that, right? So that kind of the homology methods is also utilized. This is the summary of the combination approach, in which we are just combining the two approaches together. So this all are the CGC kind of the approach only. These are the different kind of the approach based upon the assumption that coding sequences are more conserved than the non-coding region. You can have the two approach, one intragenomic and one intergenomic. Intragenomic means we are just finding the gene families, and intergenomics means we are also comparing the cross species comparison of that. You can also have the alignment of the homologous region. It is difficulty to define the some lipids. So this part is coming in the comparative genomics. Again, we will have the full lectures for that later on in the detail. So this is the summary of the extrinsic approach. Extrinsic approach, it will rely on the accumulated pre-existing biological data. Thus, it should be produce the biological relevant prediction. Weakness, it is limited to the pre-existing biological data, error in the database, and difficult in find out the limits of the similarity. So what does it tell that? Suppose I'm getting some new sequence, then I'll be just comparing the information which already I'm having, or comparing in the database which I'm having, and depending upon that only I will find it out. That for example, if you are knowing the English language and some words are already known to you, suppose I'm speaking some word out of that, then you are able to understand the meaning of that word, you are able to understand the meaning of that sentence, because that things or that information is already available to you. But suppose I'm speaking some new word which is not available with you, then you are not able to understand. So the same way that is there in the extrinsic approach. Whatever information is available, you are able to decode it, but the same information is not available in the database, then you are not able to find out the information for that. Then we are also having the ab initio approach. In the case of the ab initio approach, what will happen? We are not using any database or any other thing. We are just using the input, a string of the DNA as the input, and whatever the output is coming, that will depending upon the different kind of the rules we have created. So that will just find out the gene finder. For example, this is a simple sequence. Initially, what I'm doing is I'm just finding that where is the initiation codon and where is the termination codon, and you can just mark out that this is the one gene which is starting from this initiation codon and it is ending with this termination codon. So this kind of the method are known as the ab initio gene finding method. It will using the only sequence information, identify only the coding exon, protein coding genes, transcription site, 5 days, 3 days you are, or it is getting ignored, integrate the coding statistic with the signal detection. So like that, depending upon the some kind of the sign, it will just find out the things in your sequence, right? Then we are also using the codon statistic. So this codon statistic is again similar to that, that whatever the codon it utilized, that would be specific for the each species. So depending upon the codon statistic also, it will be able to find out that this is having the part of the genome or not, right? So the same kind of the codon statistics are there, that is codon usage, hexamer usage, GC content, and composition bias between the coding position and nucleotide periodically. So this would be also utilized in the method. This is an example of the coding sequence statistic, and this is the human codon usage pattern. So you can see that for the glycine, you can have the different kind of the code, but out of the each code, which code is utilized at a higher frequency rate, that you can make out, right? It might not be readable, but normally for the glycine you can make out there are 4 5 different codes. Out of the 4 code, some codons are utilized in a higher frequency with comparison to the other. So this is the entire comparison of that signal sensor. That suppose how it is utilized, that for example I'm having the one entire fragment of the DNA. So in the fragment of the DNA only, the computation will find out depending upon the promoter database that where is the promoter, where is the transcription start site, 5 days UTR. Then depending upon the intron and exon database, it will find out that where is the acceptor site and where is the donor side for that, and depending upon that only it will find out that this is the exon and in between this is the intron. And at the end also you can see that there would be some kind of the stop codon, then you can have the 3 days UTR untranslated region, and at the end we are having the poly A tail. So like that only you will able to find out the entire genome structure without comparing in the database. We are just comparing the information of the promoter, we are comparing the information about the donor and acceptor site, or the stop codon and the poly A tail site. So this is known as the signal, and depending upon the detection of the signal only, this fragment is identified for presence of one kind of the gene. Suppose you are taking the signal sensor in more detail, then various pattern recognition methods are utilized. It will identification of the gene signal. It will also identify the consensus sequence. Consensus sequence means sequences which are very common. Weight matrix, weight array, decision tree, hidden Markov model, neural network, and lot many other. So many softwares are there. Each software will have their own kind of the example. For example, we are taking the example of the consensus sequence. So you can see that obtained by choosing the most frequent base at each position of the multiple alignment of the subsequent interest. So you can find out that the consensus sequences that would be having always like this. That for example, we are just using the different words melon, mango, money, sweet, cookie. And suppose you are comparing all of this, then you can just find out that aim is very common, O is again a common, and you can have the N, E, and Y. But suppose you are taking only this information, then what will happen? You will just find out the word money. But actually, in the real sense, so many other things are also written. So that is an example of the consensus method, that it is not always working good. So depending upon the consensus, if you are finding the things, then always it is not right also. So these are the more detail of the position weighted matrix. So you can just finding out the different kind of the matrices, that is the usage of A, G, C, and T, and depending upon the frequency it is done. So that is much more having the computational part in that. So we are not going in much detail of that algorithm part, but depending upon that only statistically it will get validated. Now again there is one more model which is known as the hidden Markov model. So this example is like that, that for example somebody is asking you that there will be rain outside or not. In the month of, suppose for example in month of February somebody is asking, then definitely you can tell that there will not be rain. But suppose the same question is asked in the month of July or August, then what will happen? You will answer the question depending upon the outside sunlight, depending upon the humidity, depending upon the cloud. So depending upon all this, you are just find out some kind of the probability, and you will tell that there would be rain outside or not, or what are the probability of rain after this 10 15 minutes or 30 minutes or 1 hour, whatever it may be. So what is happening is we are using some kind of the direct evidences, and depending upon that we are predicting the things. Same way, suppose you are just closed in a room, you are not allowed to see something, whatever is outside, you are not allowed to see anything out of the room. And suppose somebody is coming, and you are observing that the person is coming with the sunglass, and if the sunglass is black, then you can make out that whatever the person is there, that is coming from the sun outside. So outer side bright sunlight is there, so it will tell that rain will not be there. Suppose somebody is coming with the umbrella, then you can tell that at least rain is there. But suppose your umbrella is wet, then you can tell that okay, the chances of getting umbrella wet, it is due to the chances of getting rain outside. Same way you can have the so many indirect observation also. So depending upon this indirect observation, you can predict the things. Same way, such kind of the things are also done with the computational statistical method, and that is included in the hidden Markov model. And this hidden Markov model that will also see the probability of the intragenic or the region around the start codon, coding region, and region around the stop codon. And depending upon that only, it will find out that this will be your gene structure or this is the portion of the gene. This is the state of art AB initio gene finding technique. In AB initio gene finding technique, the coding sequence and signal sensors are integrated for the overall gene modeling. It will be using the machine learning techniques like the HHM and so many other. It will also utilize the dynamic programming, and depending upon only it will capable of predicting the gene of both strands simultaneously, partial or multiple gene in a sequence, and even the suboptimal exon also it will be able to find out. These are the example of some gene finding software. Some of this will be using in the practical session also. These are the basic problem of the ab initio gene finding. It is quite difficult because genes are separated by large intragenic regions. Genes are not in continuous but split in a number of small coding exons separate by the non-coding introns. In human coding sequence comprise only few percent of the genome, an average of the 5% of the gene. So we are having the lot of non-coding region also. Then the sequence signals that are essential for elucidation of the gene structures are degenerated and highly unspecific. You can also have the alternate splicing technique. Alternate splicing technique means from the one stage of the DNA with the alternate splicing, you can have the generation of more than one gene. Then even in repetitive elements, that is quite high in the mammalian, specifically in the human, 50%, more than 50% are the repeat elements, and some genes are coding regions also. Problem with the ab initio gene finding is that there is no biological evidence. No long genome sequence may have the false positive prediction, and prediction accuracy is high but not sufficient. So that is the basic limitation of the ab initio gene finding method. Now people are using the integrated gene approach. In the integrated gene finding approach, what will happen? The program will integrate the similarity search with the ab initio technique. That, for example, we will be using the genome scan method and fGNSH and the ProCOS together. And depending upon the result of all of this, that will be merged together and we'll be finding the result out of that. We'll see the one example of this also. This is the combining program of the prediction. We'll see the example of that. That, for example, with the help of the gene scan, in my genome, I'm getting the total genome of around 624 plus 111. Suppose I'm using the hidden Markov gene based method and I'm getting some 624 and 91. When I'm comparing the gene which is predicted by these two methods, I'm finding that 624 genes are common between both of these. So whatever the 624 genes are there, that is precisely identified by two methods. So that would be more accurate with comparison to the remaining 111 and 91, right? So this way genome genes can predicted the 77% of the exome correctly, and hidden Markov model that has corrected the 75%. But when we are taking the both of this, they're able to identify the 87% of the genome. So that way, this 80% is quite good number. So that is the reason that we are just going for the combination methods. So I think today we'll stop over here only. In next lecture, we'll just go that after finding the gene, how we can go for the combining the information for the genome annotation, right?